Hello, my name is Jennifer Lippincott-Schwartz, and I'm a cell biologist at the National Institutes of Health in Bethesda, Maryland, in the program of Cell Biology and Metabolism in the Institute of Child Health and Human Development. Today I want to talk to you about breakthroughs in fluorescent imaging techniques that are revolutionizing biology and medicine. These techniques are based on the use of fluorescent proteins that allow the tracking, the visualization, and the quantification of molecular processes in living cells as well as whole organisms. Now, in order to visualize molecular events within cells, which are about anywhere between 20 and 50 times smaller than the smallest things that we can see with our naked eye, it's important to use light microscopy or light microscopes. These are all built on the principle of a lens magnifying an object. Now, if we peer through one of these microscopes, as shown here, at cells, what one sees is a beautiful display of these cells, but in a monochromatic fashion. One can't discriminate with just this black and white image what are the inner workings or the constituents of these individual cells. And so that has led to a challenge among biologists to try to decipher using alternative approaches at the constituents inside these cells. And one strategy that has been proved very successful is the use of light, in particular light from living organisms. Now, light given off by living organisms is what is referred to as bioluminescence. This is the ability of organisms in response to an activating signal, give off light. And this is characteristic of many organisms under the ocean. In fact, 90% of all marine organisms have bioluminescent capacity. In this sea firefly here, you can see that it's only particular parts of the organisms that are giving off light, bioluminescence. And that's been the key to the strategy of biologists to try to employ this technique to visualize living cells and organisms of choice. But let me give you a little background about this bioluminescence. Humans have known about it for thousands of years. In fact, the Indians in the Andes used to take glow bugs, put them between their not too different from these sea fireflies, put them between their toes to walk in the woods at night. As recently as World War II, the Japanese prepared thousands of vials of these cyprodinia in order to rub on the backpacks of these soldiers in the jungles of Papua New Guinea in order for them to be able to track at night without being detected by the enemy. Bioluminescence has even saved people. This is a bioluminescent dinoflagellite found in the ocean, and it has the property of glowing in response to agitated water. And that property saved the life of James Lovell many years ago. He's an astronaut who was flying off of a aircraft carrier when his jet cockpit, his instrument panel in his cockpit, went completely dark. And he thought he was a goner, not being able to see anything except a very faint shimmery glow from the dinoflagellites that led him to his ship. And he was able to land in complete darkness as a consequence of the bioluminescence from these organisms. Now, by far the most exciting and recently useful bioluminescent organism that we've taken advantage of as biologists is the Jellyfish Aequorea, which not only is bioluminescent, but has a green fluorescent protein associated with it. The bioluminescent molecule is aquarin, and in response to calcium, aquarin gives off a blue light that is absorbed by the green fluorescent protein, and in response to that absorption, the green fluorescent protein gives off its own light, a green light. So the green fluorescent protein is not what we would call a bioluminescent protein. Instead, it's a fluorescent protein. And what we mean by fluorescent is the property of a substance to convert light from one color to another as a result of an electrical change in the fluorophore in response to the emitting light. So if you look at this diagram right here, we have incoming light of one color that's hitting this fluorophore, and in response, not only does it reflect the incoming light, but it can also give off light of a different color, in this case green, which if you use appropriate barrier filters can be used to screen out every other color that's given off by this specimen other than green, and therefore see the signal of the GFP. Now, to give you just a little background on the sort of atomic description of this process, this is an electron orbital diagram showing electrons circulating around a nucleus of a molecule that's part of a fluorophore. And what's happening during this fluorescent response is that when light comes in of one color, as shown here in this blue, it excites the electrons that are circulating around the nucleus and causes them to jump to a different orbital. And once those electrons are at a different orbital, they will stay there until at some point they drop down to the original station, the original orbital, and in response to that shift in orbital state of the electron, a new light is given off, in this case the green fluorescent light. So this characteristic of fluorescence is what has become an extremely exciting new tool for biologists, in particular because the molecule that is responsible for fluorescence in the jellyfish, this GFP, has been shown to be isolatable and capable of being inserted into an exogenous organism. So for instance, you can take the GFP from the jellyfish, clone the gene of the GFP from the jellyfish and put it in the mouse, and you get a fluorescent mouse as shown here. Now, the incredible scope and revolutionary implication of this discovery, which was jointly led to the giving of the Nobel Prize last year in medicine to Shimomura, who was the first to purify GFP, and Chalfie, who was the first to actually insert GFP in an exogenous organism, and Roger Chen for the development of the GFP. Now, the GFP fluorophore is situated in this beta barrel-like structure of the GFP molecule, and it's this beta barrel-like structure that gives the GFP its stability and allows it to be able to be stitched together with proteins of interest. You tag the GFP at either its C or N terminus to proteins that you might be interested within cells, and then you can express these molecules within your cell or organism of interest. Now, researchers have mutated the region around the fluorophore to give rise to a whole assortment of different varieties of fluorescent proteins. These span the wavelength of the visible spectrum, as shown here, and there are also varieties of the GFP that can switch from an on to an off, from an on to an off state. Now, to illustrate just the type of expanse of colors that you can get from these fluorescent proteins, I'm showing here brain neurons that are stained through a very interesting technique called the Brainbow technique, developed by Jeff Lichtman up at Harvard. And with this technique, they're combining different types of green fluorescent protein varieties, in particular green, yellow, and red, and allowing individual cells to stitch together groups of these different percentage groups of these fluorescent proteins to create a plethora of different colors, 90 different colors to label these different neurons. And that has proved very useful for allowing researchers to determine the different tracks of individual neurons in the complex, dense network of the brain. More simply, it's possible to express these fluorescent proteins in different cell types that are being cultured in order to identify the behavior of these cells. Here's an example where blood stem cells in red are being co-cultured with bone marrow cells in green in order to understand how these two cells contact each other and exchange material in order to drive a differentiation process of these stem cells. What I want to focus on in the next few minutes is in much greater depth is the secretory membrane system and how many aspects of this system have been elucidated as a result of these fluorescent proteins and the technologies that have arisen from them. Well, the secretory membrane system is a system characteristic of all eukaryotic cells. It is all of the membranes within this system are bilayered, and they are responsible fundamentally for allowing cells to secrete various constituents. This system is also responsible for the formation of the nuclear envelope. And prior to GFP technology, our understanding of the geography and the dynamics of this system was quite limited, relying primarily on static images, really just snapshots at different time points. So there was many questions that were very unclear. But what I want to take you through is some of the data that has really provided a new type of foundation for our thinking about these organelles. Starting with the endoplasmic reticulum, what we're looking at here is a GFP tag marker that targets to the endoplasmic reticulum. And you can see in vivid detail the elaborate reticular network that defines this key organelle that serves as the site of all protein synthesis that is devoted to the secretory system. In fact, all the proteins that make up the endomembranes within the cell are derived by initially being synthesized in the endoplasmic reticulum. Now, in addition to its role in secretion, the endoplasmic reticulum is responsible for forming the nuclear envelope. And if one looks at the nuclear envelope in this slide right here, you can see it's very much looks like it's an outgrowth or connected fundamentally with the surrounding ER. Now, what I want to show you in this little video is what happens to the nuclear envelope and the endoplasmic reticulum when cells undergo mitosis. Mitosis is a key part of every cell's life. It's how cells divide into daughter cells in order to repopulate themselves in time. And what we can see in this movie is that the nuclear envelope breaks down during this process. This is a feature of many eukaryotic cells. The nuclear envelope disassembles, and that allows the genetic material to be able to undergo a dynamic interaction with cytoplasmic microtubules. Now, what one can see clearly from this movie is that when the nuclear envelope breaks down, it's being absorbed into the endoplasmic reticulum, which itself is maintained as a continuous system. Now, if we continue to watch what happens at the end of mitosis, as shown in this next video sequence, what you can see is that the nuclear envelope reforms. And the way it reforms is, again, through a pathway that involves the redistribution, the relocation of these ER-localized components into this structure that's forming the nuclear case or the nuclear envelope. So what this data is telling us is that the ER and the nuclear envelope are intimately connected throughout all stages of the cell cycle. In interphase, they appear as discrete, separate compartments, but fundamentally they're one and the same that has just been segregated spatially in interphase. Whereas, because in mitosis, these two organelles become one and the same. Okay, well, what about secretion? GFP has played a huge role in allowing us to visualize the trafficking intermediates that define the secretory pathway. Now, this pathway involves the movement of molecules from the ER through a series of substations that define the secretory pathway. And a key substation is the Golgi apparatus, which sits in the juxtanuclear area of the cell. Now, if we visualize how molecules traffic through this system, one has the ability to define the steps of this pathway. And in the movie that I'm going to be showing you, what we're going to be looking at is one type of protein, one of many proteins that move through the secretory pathway. We've tagged this protein with GFP in order to be able to visualize it in this cell. The protein has a characteristic of misfolding and being retained in the ER at a restrictive temperature of 40 degrees. But upon shift to the permissive temperature, which allows folding of the VSVG, the molecule can then move through the secretory pathway. And that temperature shift can be utilized, as shown here, to synchronously release a population of these molecules from the ER to watch them pass through the secretory pathway. And that's what you're seeing right here in this movie. The molecules are moving, in fact, there's 20 million of these molecules that are moving sequentially from ER to Golgi to plasma membrane. Now, with this type of data, there is enormous information that can be obtained. One type of information is just quantifying how the molecules are behaving as a large population. I mentioned that we were visualizing in that last movie 20 million VSVG molecules. Now, because each VSVG molecule is tagged with a GFP protein, we can correlate the fluorescent intensity given off by a single molecule with the fluorescent intensity associated with the whole cell, and thereby quantify how many molecules we're looking at any particular time. We can further define where those molecules are distributed within the cell over time. And combining those two, we can then determine the kinetics of movement of the VSVG through the different stations of the secretory pathway, where it starts in the ER, moves to the Golgi, then to the plasma membrane, and then ultimately these molecules are degraded. Now, with this type of quantification ability with the fluorescent proteins, we can determine rate constants that define the individual steps for these molecules to move through the secretory pathway. And with knowledge of these rate constants, we can then perturb the system and define precisely where in the secretory pathway the particular perturbant is affecting the system. And this has enormous potential in various studies for trying to decipher the role of either drugs that are impacting the secretory pathway or disease conditions that impact this system. So in addition to getting insight into the transport kinetics of molecules that are moving through the secretory pathway, it's possible to zoom in at higher magnification to look at the transport intermediates that are mediating this pathway. What I'm going to show you in this movie are the transport intermediates that mediate delivery of cargo from the endoplasmic reticulum, which you saw was this diffuse network, reticular network that extends throughout the cell, to the Golgi apparatus, which is situated in this juxtanuclear region. Now, the transport intermediates that are mediating this collect cargo that's synthesized in the ER at diverse sites along the surface of the endoplasmic reticulum. And as you can see, the cargo that's collected in those sites is actually concentrated. So there's a concentration step as molecules move from the ER into transport intermediates that ultimately will carry that material into the Golgi. But how does that happen? And by visualizing this process, researchers have been able to show that these transport intermediates are tracking along microtubule tracks into the juxtanuclear region where they can fuse with the Golgi apparatus. These transport intermediates are small but pleomorphic, And what we mean by pleomorphic is that they have unusual shapes. They can be long or fat, and as they track along the microtubules into the Golgi region before fusing, they undergo these shape changes. Now we can also look at the movement of these molecules once they've been situated in the Golgi, where they undergo modifications, carbohydrate modifications. That's the function of the Golgi. But as you can see in this movie here, these transport intermediates move in the opposite direction out from the Golgi in order to reach the plasma membrane. Now these transport intermediates are moving along microtubule tracks just as the ER to Golgi transport intermediates are doing. And they're moving out to sites on the plasma membrane where they can undergo a fusion reaction. And again, they're moving along microtubules as well, but in the opposite direction along microtubules. Now, what these studies have shown for the first time is that the transport intermediates that move from ER to Golgi, Golgi to plasma membrane, are pleomorphic structures that can take on unusual shapes. And these structures fundamentally use microtubules to track through the cytoskeleton, which otherwise serves as a real barrier for the movement of membrane-bound structures because of the dense meshwork of the cytoplasm. This type of information really could only be guessed at prior to our ability to tag molecules specifically and allow them to glow in the dark so that we can track them over time. Now, the Golgi apparatus is one interesting organelle that is a key component of this secretory pathway. It's involved in the modification of proteins that pass through the secretory pathway. In fact, it's a carbohydrate factory. It puts carbohydrate moieties on proteins that move through the secretory pathway and finalizes the folding state of these molecules so that once they're secreted or placed on the cell surface, they can do their appropriate function. Now, one of the things that we observed in looking at the Golgi apparatus is it has an amazing characteristic in being able to undergo a disassembly in response to various perturbants. This is a drug called burfeldin A that's been added to cells, and what you can see is that the Golgi apparatus in response to this drug completely disassembles. Now, this pathway of disassembly can be studied as a consequence of our ability to visualize these molecules in the living cell. And this pathway appears not only this breakdown pathway of the Golgi apparatus is characteristic of this organelle at every cell cycle stage during mitosis. And that's illustrated here, where we're looking again at a cell expressing a GFP tag protein that localizes to the Golgi. And as the cell goes through mitosis, you can see how the Golgi breaks down, and then at the end of mitosis, it reassembles. So this breakdown reassembly pathway previously was, although it had been speculated based on EM work, had not been able to be characterized in any significant way without the advent of our ability with these fluorescent proteins to visualize this dynamic process. So today I've just had the opportunity to tell you just a little bit about some of the things that you can do with these fluorescent proteins with respect to the secretory pathway and how they've helped clarify some deep issues within that system. But researchers in other fields have made similar remarkable findings using these fluorescent proteins, looking at the cytoskeleton, the actin microtubule cytoskeleton, looking at other organelles like the nucleus, the mitochondria. Just as one example, here's a video of the behavior of actin at the leading edge. You can see this dynamic motion here. Exciting studies like this really just underscore what GFP and its variants have provided to scientists around the world. And it's this technology that's really pushing whole new frontiers of understanding in not only biology and medicine. In the next two talks, I'm going to focus in on other types of applications of this exciting new technology. First, using photoactivation and photobleaching to highlight molecules, and finally, to use this technology to look at super resolution imaging.